Hello everyone, so my name's Luke. I lead a team called Cancer Diagnostics, which the goal of my team is to accredit or develop a new test for tumour sequencing. The purpose of doing that is to identify therapies that a patient may respond to. But to explain why we're doing this, I first need to explain to you the journey of a cancer patient currently. So, if you're diagnosed with cancer, you usually have a biopsy or surgery, and then you are treated with chemotherapy or radiotherapy with a series of follow-up. Now, the problem with this is the treatment is based on where the cancer is. So breast cancer, lung cancer, bowel cancer, for example. And when these treatments fail, options are usually quite limited. So at this point, I'd like to introduce John. John was a real patient that came through our lab. His sample came to our lab for testing. John was 49 years old when he was diagnosed with lung cancer. And it was lung cancer that had metastasized to his lymph nodes as well. So John had a biopsy. He had the usual testing, none of which was genetic. He had chemotherapy and radiotherapy follow-up, and unfortunately his treatment was failing. So I'll come back to John and explain how we analysed his sample and what we found. But I'd also like to communicate a very important message, and that is that cancer isn't a single disease. Now, we've known that for quite some time. For example, a lung cancer looks very different to a bowel cancer. Different genes drive the different types of cancers, and they respond to different therapies and have different prognoses. But what's becoming more apparent in recent years is that even tumours from the same location in the body from different patients can look very different and respond to therapies differently. So this shows you that every tumour, every cancer is unique, and that uniqueness is driven by the genetic changes that it contains. So the goal of what we're trying to do in my lab is to ensure that everyone gets tumour sequencing at diagnosis. Currently this doesn't happen, and the benefit of doing this is you can then generate a report that you give to the oncologist that will tell that oncologist what that patient may respond to. Now, we're not saying to the oncologist, you need to treat with this drug or this drug. We're just giving them more options, and hopefully, with that, better treatment outcomes for the patient. So I want to tell you about the test that we've developed here at the Garvan. We've actually partnered with a pathology company over the road called SIDPath. They are located within St Vincent's Hospital. Now, the reason that we've done this is because SIDPath are a pathology company that every day process thousands of samples similar to this. They receive tumour samples, they embed them within paraffin to preserve them, they process them by making slides, they perform all the data entry. They're set up to do this as a pathology company, whereas our expertise is in genetics. What SIDPath also do is something called histopathology, where you generate slides, and down the microscope you can have a look at what type of tumour it is, how much tumour tissue there is, and other things that tell that pathologist a bit more about the biology of the cancer. Then that sample will come to us. We do the genetic testing, we identify the mutations present, and we link those mutations to specific therapies that should work in that patient. And then we generate a report to give back to the oncologist through our pathology partners, SIDPath. So let's pick that apart a little more. There's obviously more to it than 4 stages. A request form comes in, SIDPath do the data entry, they then get the block, the tissue, from the lab where it's stored. Now that isn't trivial because there are many different pathology labs around the country, all of which receive tumour tissue and do the same thing, embed them in paraffin. And so they are set up to actually retrieve those blocks from around the country. They then assess the tissue to determine whether it's appropriate for our genetic testing, and they prepare those tissues on slides for us. They then transfer data over to us from across the road using a secure encrypted data transfer method, which is essential for handling clinical data. And then we do what my team is good at in the lab, and that is to handle DNA and RNA. We extract that from the tumour tissue. We perform the DNA sequencing. We store all that data, which is terabytes worth of data, in Garvan's IT infrastructure. We perform quality control assessment, and then we identify the mutations, link it to therapies, and produce that report. So let's go back to John's tumour. This is an example of the microscope slide that SIDPath will generate for us. So you can see with a tiny, tiny amount of tissue, we can get enough DNA and RNA to do our test. And in fact, we only need 20 nanograms of DNA and 40 nanograms of RNA, and you can see we've got much more than that. And we use an automated extraction method, which is shown on the left here,
to prepare templated ion sphere particles, or
audio associated with that, apologies. So I'll go back, you can see it working. And so we also have automated sequencing as well. So this is an example of a machine we use. It's called the Iron Chef. And so our lab is actually called the kitchen. We've nicknamed it the lab because we have an Iron Chef, and we've named all of our instruments after chefs. This one's Nigella. We have Gordon Ramsay as well. Manu. Gordon Ramsay's very temperamental. He keeps breaking down. And so after we did sequencing on John's tumour, this is what we managed to generate from his DNA and RNA. So normally anything above 5 million DNA reads is enough, and anything above 1.5 million RNA reads is enough. So we've got many, many reads. We have, across the region that we're looking at, across the genes, we have 2,300 or more reads for each gene. And there's some other metrics there. Basically, it worked very well, so that's good. Then what we do is we perform what we call bioinformatics analysis. We analyze all of that data using computers, and that's just to identify the one spelling mistake that there may be in that tumour that would link it to a therapy that that patient may respond to. And then we also use other software to link mutations to the therapies. And that's all approved therapies that you can either get through PBS or through clinical trials. So what did we find in John's tumour? Well, these are 5 mutations that we identified. Now, the genes are on the left. We have MET, PALB2, BRCA2, and the notation on the right, the mutation, is kind of like GPS coordinates for the genome. It tells the scientist which gene, where in that gene, and exactly what change was present. So that's what that notation is on the right. And the thing that stood out immediately was that John's tumour had 3 mutations in the same gene. So that's a bit of a red flag, because it shows you that that tumour is likely very dependent on these mutations. These mutations actually hyperactivate this gene. And there's also a therapy that is predicted to work in patients with mutations in this gene called MET. And that's called crizotinib. And so we come to the last stage of our tumour testing, and that is what we call an MDT, a multidisciplinary team meeting. and that consists of oncologists, scientists, pathologists, and we all get together, we review each case, we decide what goes in that final report, and we recommend what to do with that patient to the oncologist. So in John's case, we recommended treatment with crizotinib. At the time that we received his tumour, we were told that he was actually in ICU and not doing very well at all. But now he's in remission. He responded almost immediately to the drug. So that's a real success story of this type of tumour testing. So just to summarise, this is the kitchen. This is my senior scientist at work here, Electra. And what we've managed to do is develop a test that tests around 160 different genes. We test DNA and RNA. It's an end-to-end service, and it's something that we are hoping will make this type of testing available to everyone. So thank you for your attention.